We've talked about the derivation of the Michaelis-Menten equation, and we need to talk a little bit about some kinetic constants besides Km and Vmax. Now, Km and Vmax are the most important kinetic constants. However, there's some combinations of them that can tell us some important information about enzymes and let us compare them to each other to see which is the better enzyme. Now, the two kinetic constants that we're going to talk about today, one is called turnover number, right? And the other one's called catalytic efficiency. And these two kinetic constants tell us some different information about enzymes. So the turnover number is actually called kcat, and this is where it's confusing. Turnover number is kcat. Catalytic efficiency is called catalytic efficiency. It's not called kcat. So students often get mixed up, understandably, by thinking that kcat is catalytic efficiency. It's actually just turnover number. Now, turnover number suggests that we're asking how often an enzyme can turn over substrate into product. How productive is it? And so the way to gauge that is we look at how fast this reaction can go, the Vmax, and we divide it by how much enzyme it takes to reach that maximal velocity. This is a useful number. You can say, well, is this a good enzyme or not? How well is it turning over my substrate to product? Well, how fast does it go, and how much enzyme does it take to go that fast? So an enzyme that has a really high turnover number, a high kcat, is going to have a high velocity, and it's going to have a small amount of enzyme. So with a small amount of enzyme, it can get to a really high velocity, and that indicates a high turnover number. Now, if we look at this list of some example enzymes here, it indicates what their Km is. And remember, Km is an indication of how well that enzyme binds to the substrate. It doesn't really indicate whether it's turning over that substrate into product. But the kcat does, because it has some velocity information in there. And you can see there's a wide range of turnover numbers from this list of enzyme, right? Going all the way from 10 million down to a 20th, pretty much. So there's it's like a range of a billion almost, or half a billion difference in the turnover number. So enzymes are not equivalent. That's really the theme here, is that they have really, really differing abilities sometimes in order to turn over that substrate to product. They're not all awesome, right? Now, just because they're not all awesome, that doesn't mean they're not at the appropriate activity for whatever biological process they're involved in. Right? These enzymes have evolved, and they've evolved to become better enzymes, worse enzymes, or just whatever activity is appropriate for that biological process. Chymotrypsin here doesn't have a really high turnover number using this as a substrate. Maybe it doesn't need to, right? It's got plenty of time in order to digest this. This is just an appropriate level of enzyme activity. Maybe if this enzyme activity or the turnover number was higher, then it would cause problems, maybe digesting part of your digestive system instead. Now, that's turnover number, right? Vmax over total enzyme. The other number, kinetic constant, that we think of a lot and you see in the literature is the catalytic efficiency. The catalytic efficiency brings in binding of the enzyme to the substrate into this consideration. So if you look at kcat, there's no sort of enzyme substrate binding information there. It's just how fast can it go, how much enzyme's there. Catalytic efficiency takes that kcat and it divides it by the Km, which, as we know, provides enzyme substrate binding information. And so in order to have a high catalytic efficiency, you need a high kcat and, again, a low Km. A low Km indicates high affinity between enzyme and substrate, and that's going to give you a high catalytic efficiency. And again, there's a huge range between in catalytic efficiency for these enzymes. Another one to point out is this first one, acetylcholine esterase. This is involved in neural transmission. Acetylcholine is secreted between the axon and the dendrite and through the synapse of two signaling neurons, and this signal needs to be destroyed, right? So that that nerve impulse doesn't continue when there's not an upstream signal in place. And so this enzyme, acetylcholine esterase, destroys acetylcholine, and it does it really well. And you can imagine that enzymes involved in neural transmission probably have to work really fast and be very efficient, because that biological process is a really fast process. Interestingly, um, this enzyme is also a target for a lot of battle gases or chemical agents like nerve gases. You've heard of VX gas or sarin gas or soman gas. And some of these things, unfortunately, pop up in the media every once in a while. And it turns out that those nerve agents inhibit acetylcholine esterase, inhibit this enzyme, so that when acetylcholine is secreted and a nerve signal is transmitted, that nerve signal continues to be transmitted inappropriately, because this enzyme is not functioning to break it down, because it's being inhibited by that nerve agent. That causes paralysis, pretty much, not from lack of neural transmission, but inappropriate neural transmission. Your diaphragm clenches and it doesn't unclench, your irises contract, and that causes death, the diaphragm.